So today what I'm going to do is I'm going to talk about some work we did. I could actually uh, basically have named my presentation things I did during the lockdown, during the pandemic to keep me sane. But this is actually work. Obviously, COVID-19 came in 2019, the very end of that. I just like everybody, I was pretty bored, and being the arrogant bioinformatician that I am, I decided, oh, well, I can figure out this, SARS-CoV-2, by myself. And well, actually, what I did was I got some people, I knew some people at Illumina, and we actually gathered some data and made some pretty interesting discoveries on a related virus that absolutely has applications for the human condition. So that's what I'm going to talk about today. So what I'd like you to get away from this talk is, number one, I really want you to get an appreciation for utilizing outside data sources. That the amount of data that you can generate yourself is very, very small compared to the just vast terabytes worth of information out there. So hopefully, as you go through this talk, maybe that'll encourage some of you to actually go out and explore some of these outside data sources and pull them in for your own research. Also, we're going to go over different ways to maybe visualize and analyze this data. As I talk about this study, I'm going to bring up a few little key aspects. We're going to talk about genes that increase with infection, and particularly, I'm going to talk about a cytokine XCXL10. And then we're going to also talk about some of the genes we found that actually decrease with the virus. And in that talk, I'm going to talk about BEX2. So we'll discuss that. Given that the symposium is based on, drug development, I definitely wanted to put in, a section on compounds that we found that could potentially use as treatment for COVID-19. And then at the very end, I'll take any questions that you might have. In bioinformatics, everything starts with the central dogma of biology, being, you have DNA that makes RNA. You have genes in here that transcribe RNA that eventually gets transcribed into proteins, right? You have, this is just in humans, you have 6.4 billion nucleotides, approximately 25,000 genes, which transcribe almost 500,000 proteins. Just this data alone is, pretty massive as far as utilizing stuff to, characterize, say, a particular condition. But that there's obviously more that we can use, right? DNA has secondary structure. You have histones in it that can be acetylated or methylated, which would absolutely impact, the transcription of genes. Just kind of an example of this is, every cell in your body has the same DNA. It's because of this chromosomal structure that, a nerve cell doesn't look like a fat cell or a bone cell. So again, I would say the most important control on transcription, at least in the human, in humans, is absolutely the structure of that DNA. We can go to the RNA. It's not just 25,000 genes are making 25,000 transcripts, but you can also have differential splicing, alternative splicing, introns, exons. So what they've estimated, there's probably about 55,000 different transcripts that you can get from 25,000 genes. What's also been found recently, or it's actually becoming more in vogue lately, is these non-coding RNAs. And they estimate in humans, there's about 53,000 different kinds of non-coding RNAs. They can perform all kinds of different functions. And I did listen to some of the talks today, I believe one person was talking about microRNAs, an example of non-coding RNA. Again, a way we can modulate RNA expression or the expression of these genes. Again, we can measure all these. Let's go to the proteins. proteins are, it's like having a car and being able to put just about it, double headers and dual exhaust, a fancy carburetor. You can do all kinds of post translational modifications to proteins to make them very unique. Again, we can measure all of these. So for me as a bioinformatician, this is my palette that I get to choose from. These are the things that I get to figure out, what am I going to use as to characterize a particular gene? And what I want to stress and what I usually start with is probably the mRNA. The RNA seems to be reflects kind of all of this around this. It seems to be central, right? That if you condense chromosome, you condense the DNA, you don't allow transcription of certain genes, you're going to see that in the mRNA profile. If you see more proteins, typically you're going to produce more of that messenger RNA to make that protein. So for me, I really prefer looking at the mRNA to start with to try to understand a particular disease or condition or phenotype. And I have this argument with lots of people in cancer. As you can imagine, it's much easier to measure to sequence the genome rather than to get a transcriptome. So a lot of, say cancer researchers, they like the genome, but what I consider it as kind of a picture, right? You get a picture of somebody. So if I saw that maybe you saw a picture of somebody and they look like this, you might say, that's kind of a bad person. I don't want to have anything to do with them. But then say maybe you saw a movie of that person and they're singing songs to, little children. You're like, oh, that's a nice person, right? The picture doesn't tell everything. It's the movie that actually shows the character. So for me, again, when confronted with, if I was either to take genomic or transcriptomic data, I usually go for transcriptomic because, again, it's the genome in action. And these were all from Kindergarten Cop, in case you didn't know. I don't know these people. So what kind of transcriptomics are we going to look at as far as viral infection? And there's a lot going on and a lot of things that the transcriptome can tell you. In this particular case, we have a virus here. It's binding to its receptor on the outside of the cell. It gets engulfed. And once it's recognized, or a lot of mechanisms will recognize that naked RNA or DNA, and then basically start this signaling cascade, which produces inflammatory cytokines and interferons, particularly. Again, a lot of things, it's inducing cells to protect itself, but what it's also doing is it's producing things to warn its neighbors. And that's definitely what we see in these cells, is that once the virus comes, it's producing a lot of cytokines, interferons, which interacts with your immune system that causes inflammation. So this is kind of like the army coming in. It's just, lots of bombs, just kind of non-specific targeting. But eventually what happens is you produce, beta cells that produce specific antibodies against it. And these are more like the sharpshooters. But what happens sometimes with, say, viral infection is that this hyperinflammation, this signaling to the immune system just kind of goes on a loop. And this is what causes most of the problems in people, say, infected with COVID-19. That most of the people that die or are really sick with COVID probably don't have the virus in them anymore. They probably cleared it. It's this response, this hyperinflammation that is the problem. And again, if we can understand the transcriptomics of this, we understand what genes get turned on and off, that gives us the potential to potentially modulate this entire system. So where do we get this data? Where can we find some of this data out there? And one of the most common places that you would find this is in Gene Expression Omnibus. If you've never heard of this site, I would definitely write that down right now. This is where we're sticking most of our omic data, in this country and in other countries as well. If you get funded by the federal government, there is a law that makes it that you have to basically show your data to everybody, right? We're the taxpayers. We paid for this research. We should be able to see it. So for me, this is a huge playground, right? I mean, this was actually a few years back, but right now there's probably about, oh, I would say well over 5 million samples in here. And again, it's on everything you could possibly imagine, especially if you're looking at things like cancer or something like that. Lots and lots of cancer data sets in here, lots of things that are probably related to your research that you could also incorporate. Besides Geo, here's some other biological databases out there that you can collect data. One of the ones that it's kind of a recent one, it's called Refine Bio. And it's a really good place to find data. They've gone through Geo, they've downloaded everything and put it in a very convenient format for you to basically download that data, and it's well curated, which is really nice. I'm not a big fan of their search engine, so maybe if somebody at Refine Bio is listening to this, please change that. Also, you've got kind of the European version of Geo. This is the European version of our Geo, much easier to kind of work with. I did see in Oliver's talk, he did talk about R2 Genomics. Again, a really good place to not only download data, but to also do analysis on the site as well. And then I heard some talks today talking about TCGA, the Cancer Genome Atlas. Had to figure that out. Another great place to get data as well. There's also private databases where you can go. QIAGEN has their own little omic land where they've basically taken data and put it there, curated, that you can download. Obviously you have to pay for that access. You can, not only do they have like all their data, but they have little lands in here: disease land, genetics land, onco land. There is also Biobank, I'm sure some other people have heard of. This is a UK study where they've basically taken thousands and thousands of people in the UK. They test them every month, keep the good clinical records, and you can access this for a price. But today, what I'm going to talk about, where I got our data, is going to be Correlation Engine. So this is the private database, and this is from Illumina that I actually the data I'm going to talk about, this is where we got it.